All right. So, hello everyone. My name is Matt. I'm a private tutor, usually for basic sciences. And yesterday, my student was having a little bit of trouble with the Meselson-Stahl experiment. And so I figured if she was having trouble, maybe some of you guys are too. So I just wanted to go on here and kind of explain it. Hopefully I can teach you guys something. So basically the Meselson-Stahl experiment shows that DNA replication is semi-conservative. So what does semi-conservative mean? Semi-conservative basically means that you have a strand of DNA like this. So, like so, sorry. And that DNA is going to be replicated into two other DNA strands. Semi-conservative means that each parent strand is going to be used as a template for the next daughter strand. And it's going to look like this, right? Here and then here. So why does it look like this? And why is it called semi-conservative? Semi-conservative means that half of the DNA is conserved in each new DNA molecule. So here, and I'll highlight in blue, this is one parent strand right here, this strand, and it's conserved in the new daughter strand. And here's the other strand right here, and it's conserved in the other new daughter strand. So semi-conservative, half of the DNA is conserved, and then the other half is created using this parent template. So that's what semi-conservative means. So how do you discover that this is actually what happens? How did Meselson and Stahl actually realize that this happened? Well, basically they needed a way to differentiate the parental strand of DNA from the daughter strand of DNA. They needed a way to prove that the parental DNA strand is this and the daughter DNA strand is this. So how did they differentiate between these two? Well, they used nitrogen. And more specifically, they used different isotopes of nitrogen. And they showed here, and they were trying to prove this hypothesis by growing cells on medium that only contained N15 isotope, heavy nitrogen. So that means these cells, when they were building up their DNA, they only had heavy nitrogen to create their DNA. So all of their DNA would look like this. If the blue is heavy, all of their DNA would be slightly heavier than normal DNA. And then, as soon as they had enough cell replications for all of the DNA to be heavy, they immediately switched all of the cells to a different medium called N14 medium, where all of the nitrogen is now lighter. It's missing one neutron. So they switched the cells to this medium and allowed them to divide one time. So they allowed DNA replication only one time. And after this DNA replication, they lysed the cells, they extracted the DNA, and they centrifuged it down a test tube. And what a centrifuge does is it makes layers of this DNA depending on how heavy it is. And they saw that after this first experiment, they only had one band, but the band of that DNA was slightly higher. This hybridized DNA was slightly higher than the heavy DNA band that they centrifuged before, and it was also slightly lower than the N14 DNA band. So that shows that this DNA is actually a hybrid of N14 and N15 DNA. I have an image below which we can get in more detail first. So then they allowed the cells to divide again, and they saw two bands. One band was the same, it was the hybrid band, and the other band was higher, and it was purely N14 DNA. So what does this prove? How does this prove that DNA is semi-conservative? Well, here, this is kind of exactly what they did in the experiment, but now shown through images, which makes it a lot clearer. So Here is the first centrifugation that they did. And this is all 100% heavy DNA. This is when the cells were cultured on purely N15 DNA. And all of the DNA centrifuged down is right here. And it's heavy. So because it's so heavy, it's lower down on the centrifuge. And that basically proves that all of the parent DNA is N15, right? And then they allowed the cells to replicate once. And once they replicate once, all of the parent DNA is now going to act as a template for the new DNA, but all of the new DNA is now going to be N14 because there's no N15 media left, right? So that means you're going to create these hybrid DNAs. So you go from here. This is the N15 heavy DNA. And during DNA replication, you're going to split these. So you're going to get one heavy parent on each. And then you're going to have, this is N14 nitrogen in the medium. And these are going to form base pairs. And they're going to create the daughter strand, which is lighter. The daughter strand, which is lighter than the heavy stuff. And you're going to get a hybrid of parent and daughter, a hybrid of N15 heavy parent and N14 light daughter. And then when you lyse the cells, you extract the DNA and you centrifuge it down, you'll see that you'll get a band that is higher than the heavy stuff, but also lower than the pure N14 DNA. And then finally, just to further push this point, they allowed another replication. So they went from here to replicate again. And what they found was they now had two bands. They had one that was purely light. This correlates to these two DNA strands. And then they still had a band that was hybrid, and that correlates to these two. So how does this work in their genetics? Well, we can go like this, bring all of this up. And now we're dealing with, just these two hybrids. And so what is going to happen now? Well, remember, we still only have N14 medium, right? Here and here. Now all of these are going to act as parent strands. So I'm going to separate them out. One of them is the heavy N15, and one of them is the light N14 strand. They're both parent strands now. Here, one of them is the light, and then one of them is the heavy N15 parent strand. And now we know we're in N14 medium. So all of the daughter strands are going to be lighter. They're all going to be N14. So here, the daughter strand is going to be N14. Here, the daughter strand is going to be N14. Here, and here. Sorry, my DNA looks awful, but yeah, I hope you guys get the point. And this shows two of them are purely N14, and then two of them are hybridized. And that's basically what the experiment showed. So how did this experiment prove semi-conservative replication? Well, the alternative hypothesis is conservative replication. And this is what it would look like if it was conservative replication. Here, you have your DNA. Oh, let me actually make some space on here. So here you have your DNA. And conservative replication would be, you still make two DNAs because it's replication, but conservative is you conserve the parent DNA. And then you make another new DNA that is all daughter strands, and it's all new DNA. And we know that doesn't happen because we saw the presence of these hybrids. And so that's why conservative replication is a false hypothesis, and actually semi-conservative replication is the true way that our cells and our DNA does replicate. So I hope that cleared up some things. If there's any other questions you have, please, I am definitely able to help out. I'll help out the best I can, but thank you.